Thanks, Agnes. I think you do a very great job to entertain the group here. And so people are ready to listen to the next session. And the session I have the privilege to chair, and that's the session about genetics and breeding of biomass, or with other words, resource use efficiency. And my idea is that biomass is not everything to start a successful barley breeding program, but without biomass, no success is possible. And the art in breeding is to find the right balance between yield, quality, and biomass. So that's what we try to do. And for this, we have four speakers in this session. And the first speech or talk will be given by Daniel Julio Miralles from the University of Buenos Aires from Argentina.
Thank you very much. Thanks for the invitation by the organizers. To put in context my talking, let me show some numbers, some statistics in relation with the barley production in Argentina. This is Argentina, it's in South America, and Argentina is the sixth world exporter country in terms of malt, in terms of malts. And within the region, Brazil is the world leading importer country in terms of malting barley. And within Argentina, the harvest area of barley is around 1 million of hectares. The production is close to 5 million of tons per year. This is the last statistic from the last year. And with a national average yield of 4.4 tons per hectare. The main destination of our barley is for malt, for malting barley. We use barley for feeding, but in general, the main destination is for malt. And the crop is concentrated in the province of Buenos Aires. This is the province of Buenos Aires that concentrates 91% of the whole production. And within this area, most of the production is concentrated in the south and part in the west, and less production is concentrated in the north. In fact, in this area, barley wheat was replaced by barley during the last year. And our malting capacity at country level is 875,000 tons. 70% of the malt production is exported, mainly to Brazil. This is the main destiny of our production, but we provide malt to the rest of the country, Chile, Bolivia, Colombia, and Paraguay. And this is the distribution of the malting houses. Our malting houses are concentrated in the southeast, where the crop is concentrated, and there are some malting houses in the north. I show you how was the dynamic of the harvest area in the last 40 years. The harvest area of barley changed during the last 40 years. From the 60s to the 80s, the area designated to barley was reduced. And after that, from the middle of 80 to the middle of 2000 was increased a little bit. And during the last 20 years or 25 years, the area designated to barley was increased a lot. And we reached 1.5 million of hectares in some years. And at the moment was stabilized around 1.1 million of hectares designated or designated to barley crop. And in terms of yield, this is our national average yield during the last 40 years. And you can see in this figure that there was a little increase from the 60s to the 80s, around 13 kilograms per hectare per year. This is the yield gain, was relatively low. But from the middle of 80 to nowadays, the yield was increased at the rate of 64 kilograms per hectare per year. And in this arrow that you can see in this figure, at the end of the 90s and the beginning of the 2000 year, there was a very important integration or introduction of the European varieties. In fact, nowadays, most of our production of multi-barley is concentrated in European varieties. And in relation with the production, the same shape that I show you for harvest area, there was very few production between the 60s and the 80s, but from the middle of the 80s to the 2000 increase, there was a light increases, and during the last 20 years there was a very important increase in barley production at country level. So this is a very briefly description of the barley production. Now I will concentrate it in the second part of the talking which were the physiological and numerical yield components that were modified by breeding in the last 40 years in Argentina. To do that, we select 11 commercial genotypes that were widely used and now used in Argentina at the moment. We make 2 years of experiment in our units. The selected varieties were released at the commercial market between the 1980 to 2019, 2019, and the cultivar were sown in both years 2020 and 2021 under the best agronomical condition without nitrogen, phosphorus, and other nutrients restriction, and without water restriction. All the experiments were irrigated. This is, you can see in the left, the year of release of the varieties and the name of the varieties that were used in Argentina in the last 40 years. These two varieties were local varieties released by local breeder in Argentina, and the rest, from Scarlet to Yanara, they are all European varieties. Maybe some of these names are familiar for some of those. And we measure the phenology, we measure intercepted radiation during the whole cycle, and we separe pre-anthesis and post-anthesis intercepted radiation and radiation use efficiency, the aerial biomass, the grain yield and the physiological and numerical components, nitrogen concentration in grains and biomass, and also the malting parameter that I will not show in this talking because I have no time to describe the whole malting parameters. This is a picture of the experiment. They are in our unit. This is the plots. We had 4 reps in the 2 years. And another picture with the levels are the year of release of the varieties. You can see there. And you can see the difference slight difference in phenology I show you. And the results show that the first results are related with phenology. This is the day to anthesis and the days from anthesis to physiological maturity, the grain filling duration at right, against the year of release of the varieties. And you can see that in both years there was a positive relationship between the days to anthesis and the year of release. In other words, the breeding enlarged the cycle of the varieties up to anthesis. In both years, the differences between years in red and blue are as a consequence of different sowing dates. In 2021, sowing date was early respect to 2020, and this is the difference in the duration in calendar days. And there are not very different or important difference in relation to the year of release during the grain filling period, but there was some difference between years. And when we measure the same duration, pre-anthesis and post-anthesis in thermal time, for eliminating the effect of temperature during the cycle. There was a common line of regression, but the trend was the same. The modern cultivars were longer respect to the older ones. In other words, again, the breeders or the breeding enlarged the cycle of the modern varieties respect to the older ones. And when we measured the duration of the grain filling in thermal times units, there was not an important difference between all and modern varieties. Plant height was reduced by breeding. In both years, you can see a negative trend when we plot the plant height against the year of release. And the rate of reduction was 0.22 centimeters per year. This was the rate of reduction in plant height. And the stem diameter was very important for us because the older varieties like this, I show you there, lodging was really a problem for the farmers. And the breeding increased the stem diameter, at least if we compare the varieties released at the beginning of the 80s with respect to the varieties released at the end of the 90s. And the modern varieties, in general, there are had not important difference in the stem of the diameter, but has a greater diameter respect to the older ones. In terms of grain yield was increasing. The modern varieties has showed higher yield respect to the older ones, and the gains was 69 kilograms per hectare per year. And if we measure the yield gain in relative values, the yield gain was a little bit less than 1% per year. This was our genetic yield gain. When we dissect what happened with the yield gain in the physiological component, the aerial biomass, and the partition to this biomass to the grain, the harvest index, we did not observe any trends in relation with the biomass. Biomass was not modified by breeding in Argentina in the last 40 years, but breeding increased the harvest index, and the harvest index passed from 0.4 in the older varieties to 0.5 to the modern varieties. This is our highest harvest index at the moment. And when we again dissect the aerial biomass in the cumulative intercepted radiation and radiation use efficiency, the cumulative intercepted radiation show a slight positive trend when we compare the old and modern cultivars. Probably this is associated with the longer cycle of the modern cultivar with respect to the older ones. But when we see what happened with the radiation use efficiency during the whole cycle, there was not any trend. It was fairly similar when we compare the old and modern varieties. And when we inspect in more detail what happened with the cumulative intercepted radiation and the radiation use efficiency during pre- and post-synthesis period, we can see that during pre-synthesis period, the old varieties, these releases in 1980 and 1990, at the end of 1919, show lower cumulative intercepted radiation. There are another modern cultivar that also show the lowest intercepted radiation, but in general, the modern cultivars, especially these cultivars, show greater values of cumulative intercepted radiation. When we observe what happened with the intercepted radiation during the post-synthesis period, there was not a clear pattern between old and modern, probably because there are not important differences in the length of the cycle. And I have not the data here, but there are not very important difference in the leaf area index between the varieties. And the radiation use efficiency, we observe that in this modern variety that are the most popular varieties at the moment in Argentina are Andrea with this, and Charles is the other variety, had the highest radiation use efficiency during pre-anthesis. And there was, again, not a clear pattern in terms of the radiation use efficiency during post-anthesis and during the whole cycle. When we dissect the numerical components of the grain yield in the grain number per unit area and the average grain weight, we can see in both figures that breeding increased both grain number per unit area and also average grain weight, which is was a surprise for us because in general there are some compensation between the number of grain per unit area and average grain weight. But in this case, the modern cultivars not only has more grains per unit area, has or at least maintain or a little bit increase a little bit the average grain weight. And this is the reason why there is a relationship between grain yield and grain number per meter square and grain yield and average grain weight, at least up to yield of around 8 tons per hectare. These both axes are in grams per meter square. If you multiply by 10, you obtain the kilograms per hectare. And from 7 to 8 tons per hectare, there was a clear linear relationship, but from 8 tons or high yield, there was not a clear relationship between yield and grain number or between yield and grain weight. And again, we dissect the grain number in the spike per meter squares and grain per spike. And when we observe what happened with both yield components, there was not a clear relationship between the between the number of spike per meter square and the year of release. In other words, the old and modern variety has or had the same number of spike under those potential conditions was the same, exactly the same. But the modern varieties, at least from the older ones that were released in the 1980 to the 2000, showed an increase in the number of grains per spike. And this number of grains per spike was maintaining in the varieties released from the 2000 to nowadays. This is the circle described where we observe the greater increase in the number of grain per spike. And what happened with the grain protein? Because it is expected that if you increase yield, probably there will be a dilution effect and the grain protein could be reduced as you increase yield. But in this case, at least in that conditions and in both years, the grain protein was maintaining when we observed the old and new varieties. This is the industry requirement to when you sell the malting barley to the companies. And in general, the grain protein was maintaining during the different years of breeding. And the nitrogen harvest index was a little bit increased. Again, from when we compare the varieties released between the 1920 to the 2000, there was an increase, and in the whole modern variety, the values of the nitrogen harvest index was maintained. So, some conclusions: breeding increased the length of the cycle. In fact, our modern cultivars that are used by the farmers are longer respect to the older ones, especially from emergence to anthesis. For us, anthesis is when the awns appear over the flag leaves. This is the real anthesis time, similar to the anthesis in wheat. and without change in the duration of the grain filling. This is an interesting point because we used to use our rotation wheat and soybean or barley and soybean. And if our new cultivars are longer, we have a delay in the soybean in the next crop, which is normally soybean. And soybean yield is reduced at the sowing date is delayed. And this is important to put the new varieties a little bit later than the older ones in the field. The other point is that the yield was increased at the rate of 69 kilograms per hectare per year and represent around 1% per year. And if you remember one of the first figures that I showed in the presentation, our national average yield in terms of yield gain was 64 kilograms per hectare per year. Both numbers suggest that the most national yield progress in Argentina was associated with genetic gain, mostly, unless with the crop management in the field. Crop management, I refer to different sowing dates or different nutrition characteristic in the crop. And when we're exploring the physiological components, genetic gain was associated with a higher partitioning to the grains, more or higher harvest index, because biomass was not, or vegetative biomass was not related with breeding, at least for this set of varieties. And the cumulative interceptor radiation and radiation use efficiency during the cycle was not modified in the important manner. The yield numerical component showed that genetic yield gains were associated with increases in grain number per meter square, as well as in average grain weight. In other words, breeding increased grain number, but also maintain or little bit increase grain weight, which is very important for the industry because grain weight is related with grain size, and grain size is one of the parameters very important for the industry. And the improvements in the grain number per unit area were a consequence of the increases in the grain number per spike, because the spike per meter square were not modified. And despite of the increases by breeding in yield, the protein was maintained, which is a good signal for the production, because the dilution effect was not clear, at least in this data set. Many thanks. This is some picture of our units, and many thanks for your attention.
Thank you very much, Daniel, for your progress report on breeding in Argentina and manifestation of improvement of barley quality and yield. You didn't mention if barley is exclusively used for brewing or also as a feedstock.
Not exclusive, but most of our production go to the industry for brewing, for malting barley, and a very few proportion go to feeding. And there are, at the moment, which is a very important point in the future, we don't produce barley for human food in general. It's not very common in Argentina.
Thanks. I open the discussion.
Yes. I want one question. Maybe you said at the beginning, but maybe I missed it. So were those varieties developed by Argentinian breeding programs or were international varieties?
No, we had a very important breeding program until the end of the 90s. But from the end of the 90s to nowadays, all the varieties that are grown in Argentina are integration from Europe or European varieties from different countries, mostly from Germany and other origins. Yep.
Next question.
Daniel, what is the driver for the longer, but breeding for the longer and growing period, especially?
Breeding for, sorry?
From the data you showed, from emergency to the flowering or heading, the time to become longer. for the new varieties. My
how
question is,
longer? Okay.
What is driver for that?
Respect to the older one, you mean, or the longer of the cycle?
I think from your slide, the time from the emergency to the flowering time is become longer for the new varieties. The reason I ask this question is, in Australia, exactly reverse. Yeah, my question is, what is driver in Argentina for increased yield with the longer vegetative growth period?
Yeah, the longer varieties has a little bit higher yields in general, because you can. What I mean is, it is clear that there are not important differences in biomass, but the longer varieties probably allow you to increase the number of grain per unit area. But to capture this advantage, you have to put the new varieties a little bit early in terms of sowing dates. Because if not, you put the grain filling under not very suitable condition associated with the heat shot. This is the point. Yeah.
Okay, next question, the last row in the audience.
It's on? Oh, thanks. Thanks, Daniel, for the presentation. It's more like a comment more than a question, but it's important to know for the public that probably the main driving factor in the inclusion of the European materials is the malting quality. I mean, they have been also, the industry has, in Argentina and in Uruguay, has the malting industry had to, in some ways, make the use of European materials something that they want, because there was a big jump in quality. I mean, if you have put some malting quality data comparing with the old varieties in all Argentinian varieties, that difference would be much more striking than with the agronomic traits. So in some aspects, even they improved the yield, as you mentioned and you show. There was a side effect, and a good side effect, a very good side effect. Perhaps you can elaborate from that.
The jump was in yield, not in quality. The quality of the oil.
No, I mean the malting quality.
All right. Yeah.
That's what is really interesting for the malting industry, and that differences were great. No, in the physical quality is similar.
Yeah.
Okay.
Yeah, it's a short question. So if barley cultivation is increasing so much, does barley push out something else, or you open up new land, or what is happening?
I showed that most of the barley is concentrated in the southeast. This area in the southeast was dominated by wheat in the past. But when we arrived the new varieties, the European new varieties in the last 20 years, we increased the yield around 20%. And what happened? That the farmers, there are another political problem, that we had restriction with the exportation of wheat. 10 years ago, the government decided to limit the wheat exportations, and the farmer decided to replace wheat by barley because they can export it freely. And the best region of the highest yield potential is in the southeast, and the new varieties increase the yield a lot in that. Under non-irrigation, for example, we can obtain 8 to 9 tons per hectare without irrigation in that area. And barley was replaced by wheat. And this is the reason that barley was concentrated in that and increased the area a lot, because there are not restrictions similar to we had in wheat. Yeah.
Okay, now the very last question, and short question, short answer, because we have to continue.
Thanks, Daniel. Very nice presentation.
Where is?
Here. I just wanted to ask you, you said that, okay, you cannot push forward the maturity date because you have the soya planting, but in the context of climate change, that you expect warmer summers, drier summers, would you suggest to keep on the same maturity you have now, or would you start shortening but keeping the quality?
In the whole in the whole countries in South America, the problem with the temperature is not the maximum temperature; it is the minimum temperature during the night. the nights will be warmer and warmer. And this is the driving force that increase the average temperature. And we suggest at the moment with that varieties, move the sowing dates. This is the strategy in terms of crop management to avoid this increase of high temperature, because our prediction using simulation model, we have some published paper around that, it is 7% of reduction, between 6 to 7% of reduction per 1 degree of increase of night temperature. And to avoid that, in barley and wheat are similar, the reduction, more in barley than in wheat, according with the models. And we suggest to the farmers move the sowing date early to avoid that. And put the crop in the same frost risk. Because if you maintain the sowing date, your frost risk is lower now than in the past. And this is the point. And this we suggest to move the sowing date early. And you can maintain the high potential yield with that strategy.
Yeah, Lana sent her apology. She got sick yesterday. She has asthma and probably something in the air like pollens or whatever. Anyway, she cannot speak today. This is a work of our student. So obviously we are both involved, and I think I'm well in position to entertain you for another 15 minutes. So, not much introduction about why potassium. Potassium is important. It's essential nutrient, one of the major free ones. So plenty of different functions, over 70 enzymes controlled by potassium. So obviously it's very critical for plant. We're talking about roughly 5% of a dry matter made of potassium, 3 to 6, I would say, on average. And therefore, it's very essential that barley has sufficient amount of it. So the use of potassium in agriculture is increasing as we speak. You can look at the trends here over the last 50 or 60 years. It's going up and up and up again in different countries. In Europe, it's probably the most in demand. And it's hardly surprising because we really try to make the intensive production of crops, and therefore they all need potassium. Importantly, in fact, we increased the supply since 60s, Green Revolution, probably by 3 or 4-fold. I don't have exact numbers for potassium, but for nitrogen it was 7-fold increase, really. So I think potassium is probably close to that. Why potassium efficiency is important and how it's done. Essentially, we're talking about how responsive are plants by increasing the yield when you give more potassium. So it's essentially their amount of the grain yield or whatever biomass, if you go for feed barley, for 1 kilogram of potassium in the soil. And here we're talking about the two components which control that efficiency. It's uptake efficiency and it is utilization efficiency. So there are many different factors here involved. It's root architecture, obviously, and ability to scavenge or to go through the soil and get potassium in when conditions may be not necessarily favorable. It's really the density of root hairs and length of roots and angles and many other things. And also, of course, it's expression of different potassium transporters for radial transport, for xylem loading, transfer to the shoot, and remobilization from the flag leaves to the developing grains. So there are many control points or checkpoints in this path. And when you want to improve potassium use efficiency, you really need to look carefully at this and decide which one is more important in particular environmental context. Attempts to modify the root architecture for nutrient efficiency through genetic means from marker-assisted selection failed to a large extent from what I know, because you end up with probably half a dozen or dozen minor QTLs, each responsible for 1 or few % of the total trait. So obviously it's become a nightmare, and in terms of the practical dealing, and results are not that great. But while we are talking about this, what is special about the potassium use efficiency today as we speak, why it suddenly become on agenda? There are two reasons. One is economic one. So we're talking about the increase of potash, I use here as an example, is one of the major source of potassium fertilizers. And the price is going up and up and up. Well, it was relatively steady for some period, and since 2003, it went up. Was a small drop in 2008 when we had the economic crisis, it's high again. So the total cost, economic cost of potassium fertilizers is really very massive now. And again, we have other constraints which we have here on economics. So farmers really looking for varieties which will be more responsive to potassium fertilizers and will require less potassium to be added to the soil. And here we have an issue because when we selected plants for their responsiveness to fertilizers in the 60s when the Green Revolution started, we did it under optimal climate conditions. There was not really much temperature, night temperatures, was much less global average temperatures, and we didn't have these severe drought events as we have now, or flooding in parts of the world. So essentially varieties which were responsive for nutrients, phosphorus, potassium, nitrogen, any actually nutrients we had, well, but NPKs are major ones, they are not really responding as well now under stress conditions. So the nutrient use efficiency under control and stress conditions are very different, and therefore not necessarily that elite variety, which we are dealing with from 60s or 70s, will be a good one responding in the field now if the stress is an issue, and it is an issue in many cases. But also important thing is eutrophication or the environmental consequences, and this is really the result of this poor ability to respond to fertilization. So we try to maximize productivity by putting more and more input, and the nutrient is simply sitting in the soil and is not taken by plants. Essentially what happens is just end up in waterways, in lakes, streams, and you have these environmental issues. Again, a lot of I can talk more and more about this. It's not specific to potassium, it's relevant to all nutrients, nitrogen, phosphorus as well, so we're talking about three major ones, but that's a reality. So to have our productive systems sustainable, environmentally friendly, and economically viable, we need to keep the nutrient use efficiency in place. Potassium just happened to be my sort of favorite nutrient just historically for 20 years, but just a few months ago, I got the grant to deal with phosphorus, the same issue, and we use Tibetan wild barley collection because it's very, very efficient in dealing with the low phosphorus in the mountains in China, and we are hoping to find some interesting things. So maybe in the next meeting or meetings, I'll be able to talk about that. But today it just happened that it's historical work of a student. So now to practical results, next 5-6 minutes. What we did, we screened 30 different genotypes of barley. And again, collection was pretty broad all across the globe. We did not really lock ourselves to a particular region. 2 or 6 rows of barleys, very different varieties from different places. We gave 4 treatments, so it was really hydroponics, sort of semi-hydroponic experiments, because we wanted really very, very low level of potassium, just 2 micromole is the lowest. So we used a sand culture and modified nutrient solutions. So we gave 4 potassium treatments. 2 millimole we consider as optimal one. We have luxury supply 20, and we had the 2 deficient concentrations. And we went through the series of physiological and agronomical measurements: biomass, plant height, number of spikes, thousand seeds weights, grain yield, blah, 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 blah. So everything we could measure, again, in physiology included photosynthesis, transpiration, stomatal conductance, nutrient content, again, the whole bunch of that. So we did the cluster analysis, and we computer grouped the varieties into the 3 major clusters, depending on the responsiveness to fertilizers. Again, we did later correlation analysis. So I think this graph is probably interesting to see. So you see that this is what we consider optimal concentration. So if you reduce it to just 20 micromoles, some of the varieties are pretty happy and they are stable here. Some start to drop, and again, this is a grain yield, so it's probably 20-30% decline in the yield. But some of them really were pretty bad at the beginning and not responsive to fertilizers. So that's interesting, and it gives us some hope that we can understand the genes behind that if we look at the physiology of this. But this is about the grain yield, and when you look at the shoot dry weight, you can see there is absolutely no difference in plant responses to potassium fertilization. So obviously that brings a sad message to us. You cannot use the growth response, biomass of the shoot, as a potential indicator or physiological marker for the potassium use efficiency. So it's inconvenient. You need to wait until really the final harvest and get that to understand this. It's not really result we expected. And in fact, we also measured the flag leaf and everything else, and that's the same thing. Biomass does not correlate strongly with the potassium use efficiency. So this is illustrated here. So again, the major finding is a poor predictor, if you look at that. So what we did next, we look at the actual numbers of different varieties. And you can see, and this is a positive thing, you can see that they are really very different in their responsiveness to fertilization. If you take the variety flagship, the yield at the lowest, 2 micromolar, was pretty high, and it's only doubled when you give the optimal supply. So this variety is pretty happy with almost no potassium in the soil, or in a growth media in this case, and just barely increase it, well, not barely, twice, but still. And this one had almost no yield and it really increased it like 50 times. And we're much more responsive with a proper supply of potassium, nearly twofold compared with this one. That's a good thing because we have enough genetic variability amongst the varieties we use now to look at the genes and to look at the mechanisms which are in them. So we tried to look at some of the possible associations. Again, the correlation and the causal relationships are two very different things, so I will be very cautious about doing any conclusions from PCA analysis. But you can see again that these three groups in different colors, so they're really clustered in interesting ways. Some were associated with the spike numbers and grain weight, and others were basically with tiller and a shoot biomass. So it's interesting. We need to digest this information, and we need to look at the possible association with the tolerance mechanism. But so far, from agronomical part, we understood that, okay, there are varieties which are highly responsive and very productive, and there are some which are not responsive and very least productive at the low levels. So here are a few specific names. So basically, the next step for my colleagues will be to make the populations from them and to look at the QTLs. Hopefully it will be not too many, but again, I'm not sure if it's root or something else. But the most important question was what is behind these differences and what are possible physiological mechanisms. So to get some insights into this one, again, I'll skip long story because it's an entire thesis of a student. I just show a couple of slides and a couple of things. So we look at the leaf, flag leaf potassium content, and we look at the xylem sep potassium content. And when we say xylem sap, you just chop the shoot, put in the shell in the bomb, and squeeze it. So obviously, I think practically it's a mix of phloem and xylem in a specimen. What we found here really that we have a positive correlation between the xylem sap at a low potassium supply and negative at high potassium supply. So there is a feedback control of an uncoupling between the root radial uptake and long distance transport of potassium, and supply to a shoot under low potassium conditions become the most critical. So that's emphasize importance and essentiality of the long distance transport versus the uptake by roots and radial transport. And that's interesting and important thing is. And when we look at the photosynthetic performance and look at the stomatal conductance and FVFM and chlorophyll content, we found again they are really poor predictors, again, sort of negative results because SPED is such a nice thing, you press a button, got the number, but unfortunately it does not correlate. So what does it mean? It means that the phloem transport becomes central. It's really the last step in a all long path, movement of potassium from the flag leaf to the developing grains, which is critical in this process. And to study that, it's not a trivial task. We don't know too much about this because it's the most difficult part to study. I can measure hundreds of varieties and uptake kinetics by roots. I can measure easily loading of nutrients in xylem by using our techniques, but getting access to phloem is somewhat tricky. What we know from literature and what we know from other things is that it's CAT or AKT2 transporters which are critical. So AKT2-3 to be specific is the phloem-associated potassium channel which really can operate as inward and outward rectifying channels. And it was shown to control the nutrient transport or potassium transport to the grain, and it's regulated by auxin. I presume it will be also pH sensitive and have the same properties as our potassium shake channels, and it's operating both potassium influx and efflux. So overall, there are 75 possible candidates in Arabidopsis genome 40 of them classified as non-selective cation channels, and 35 are more specific potassium transporters from 5 groups. But this is a likely candidate from studies on Arabidopsis. How can we get this information? And this is what should be done in the future. As I said, the hope for QTLs for the root traits are negligibly small based on experience with phosphorus and nitrogen. Measuring the shoot biomass, SPED, chlorophyll content as predictors also will not work. Waiting for the grain yield give you the answer which variety is responsive, but again, the QTLs are sort of questionable. If that's a QT2 transporter, how can we study it? How can we screen varieties for that? And here we need to combine 2 techniques from my point of view. Again, continuation of my talk yesterday, we need to look at the transport from specific cell tissues and how we can get that. We need really to label the phloem with some fluorescent markers and use the fluorescent activated cell sorting. So essentially what I'm saying, what we need to do next, someone in this audience just give us barley varieties with phloem labeled, particularly marker, and we go for this FACS. We separate phloem tissue from protoplast or cells from phloem from others, and we can really screen them for kinetics of potassium uptake using our microelectrode techniques, and we can really do the DH lines that will give you 1 QTL. So my point yesterday was 1 protein, 1 QTL, and here will be no doubts it will be just one of them. It's not the trivial task. The cost of machine is really $600,000. So obviously it should be the team effort between the breeders and scientists. So potassium is a good physiological marker, as I said, and I'm pretty certain we can do the same thing. And I think I'll probably, for the sake of time, skip the last 2 slides about the link with salinity. So conclusions, barley accessions really differ drastically in their responsiveness to fertilizers, potassium fertilizers, and the contrasting varieties have been identified and can be used now in just traditional breeding or from marker-assisted selection. The shoot biomass is poor predictor, so we need really to look at the phloem transport and the cell-based phenotyping and developing the combination of fluorescence-activated cell sorting and electrophysiological tools can help here. Thank you.
Thank you for the presentation. I'm not sure to have seen things very clearly, but in one of your slides, you show that the yield increased with the amount of potassium in the xylem or phloem at very low potassium availability. But at very high potassium availability, if I saw it well, it was the other way around.
Yeah.
Is there any explanation for why having more potassium in the xylem will reduce yield?
That's an interesting question. From what the textbook says, like Marshner is a classical book we use for teaching, 80% of potassium is reduced back, which was transported to a shoot, is normally reduced back to the root for negative feedback control. So there's uncoupled radial transport and loading, and what I would believe here is a modification of source-sink relationship. So the high level of potassium in the xylem will probably stimulate the vegetative growth on expense of the transport of a grain. That's my best explanation. It's counterintuitive, I completely agree, because we expect at least flattening rather than negative, but this is what it is. And the 30 sample size of 30 is pretty good enough to be statistically significant. Yeah. Again, probably one comment. It also depends at what time, particularly. It's one snapshot of the ontogeny, right? So maybe 2 weeks before, 2 weeks later, it will be different.
Sergey, you focus very much on transport of potassium, let's say during through the phloem and xylem and importing into the cell. But how about the uptake of potassium from the soil and the support by the microbiome?
Microbiome is definitely critical, but I think the overall the role of epidemial-based transporters is probably very marginal in the control of the overall productivity. It's important for the biomass. So here after, the feed barley for, or not, like if a dual purpose barley, right? So we're interested in just the biomass for animal feed. Yeah, that's perfectly. So it'll be proportional response. But when we talk about the transport to the grain and the grain yield, 1,000 weight seeds, it's really the transport from a flag leaf, which operates as a buffer, and the grain, and it's a phloem-based transport, which is critical. So it can improve the first 3 or 4 components as long as you want. That will be the weak link. And if you don't target that, you will end up nowhere. I mean, you'll have a big biomass, you'll have green plants, tall plants, but the grains will be still not responsive to that.
Okay. Thank you for the nice talk. Was there any morphological study or histological study to see any association with a higher potassium use efficiency for lines which were showing higher yield?
We haven't done this work, to be honest. In the phosphorus project I just mentioned, we plan to put 4 PhD students on different components. That will include cytology and root architecture and everything else. For potassium 1, it was essentially focused on physiology and association with environmental stresses such as hypoxia and salinity, which I didn't want to mention today. So the answer is, I have no idea.
Thank you.
Thank you, Klaus, and thank you to the organized committee for giving me the opportunity to talk here at this very important meeting. So my group is interested in study photosynthesis, regulatory mechanism of photosynthesis, and molecular mechanisms that are behind the chloroplast biogenesis. We have done that for many years in Arabidopsis thaliana, and during the last few years we are trying to transfer this knowledge in barley. In particular, today, in these 15 minutes, I would like to explain you how we try to overcome one of the major limitations that photosynthesis encounter in a canopy like barley. Most of the light that is reached on top of the canopy is actually far in excess for a crop. Only 25% of it can be used for photosynthesis. All the rest is actually in excess and is responsible to cause photooxidative damages. Plants need to invest energy actually to dissipate this energy or to repair the oxidative damages. On the contrary, on the bottom of the canopy, you have leaves that are constantly under the shadow or even completely in the dark, and they do not run an efficient photosynthesis. So there are several studies that have been initiated first on microalgae and cyanobacteria cultivated in bioreactors, and later on publication appears also on crops, on model organisms like tobacco, but also on rice, on soybean, that show that you can gain in photosynthetic efficiency and in biomass production if you try to have a more uniform light penetration towards the canopy. And one of the strategies we have been using was to reduce the amount of chlorophyll at the leaf level. So if you have paler leaves, light should go penetrate much deeper into the canopy and have a more uniform photosynthetic efficiencies and in total a higher yield. This has been shown, for instance, in tobacco, where they show that these plants can even produce more if you cultivate it at higher density. This has been shown also in rice, where they show that these plants can even grow better at under highlight conditions. And in soybean, they've been even able to show that since the leaves are paler, so they have a higher albedo, so they are able to reflect more light, you have a lower temperature at the canopy level, up to 1.5 degrees less at the canopy level, and that can allow you to gain in water use efficiency, for instance. And of course, the last point is related to nitrogen. Chlorophyll contains nitrogen. Chlorophyll is not free in the leaves, it's associated to proteins, and obviously protein contains nitrogen. And there are simulations that are saying that if you reduce chlorophyll, you might have even a reduction in nitrogen input that has been estimated up to 9%. When we publish the data on the AS1 mutant, we have been also contacted by this company in Italy that is developing these plants. They are known as agri-photovoltaic plants, where you can produce renewable energy and at the same time cultivating plants. They are bifacial solar panels. They can receive directly light from the sun, but they are also working with light that is reflected from the soil. And talking with the engineer, they were asking us to try to test these plants also in this kind of systems, because by reflecting more light, there would be more light going to the lower phase of the panel and probably increase the efficiency of this system. So in other words, you produce feed and food and at the same time renewable energy. So we tried to address, as I said, this point in barley, and we were looking, obviously, for mutants that had a reduced content of chlorophyll in the leaves. And today we are using different approaches for time limitation. Today I'm going to talk you our experience with a forward genetic approach that we run essentially going to the field in one end with a fluorometer that measures the photosynthetic efficiency, and in the other end with a SPAD that is able to measure the chlorophyll content. And we have done that on two different populations. One is the Ortillus within the frame of the BarPlus project, and it was provided by Agnieszka Janak at the University of Silesia in Katowice, Poland. And the other was given to us, kindly given to us by Silvio Salvi, the Timor population at the University of Bologna in Italy. So the first mutant, what we are searching here, and that's the reason why we call these mutants happy under the sun, are obviously it's easy to find a mutant that is paler. You just have to disrupt the photosynthetic machinery and they look paler. What we are looking here are mutants that have a reduced chlorophyll content but a higher photosynthetic efficiency. And this is not common. That's why we run with the two tools searching for these mutants. The first mutant with these characteristics is actually the one that we got from the Ortillus population. You can see, as expected, it's paler, but it grows normally in the greenhouse without any problem and producing the same yield under greenhouse condition. It contains around half of the total chlorophyll with respect to the control, and it has a higher content of chlorophyll A with respect to chlorophyll B, because that's a typical data that you get when you reduce the antenna size of photosynthesis system that bind more chlorophyll B. And you clearly see that this mutant is specifically altered in the accumulation of this protein here. This is actually the region where the antenna proteins of the photosystems migrate. All the rest remain constant. In particular, I'm referring to these trimeric proteins at the level of photosystem 2 and to these dimers that we find at the level of photosystem 1. The phenotype of this mutant is retained towards the entire life cycle. Even the flex leaves looks paler with respect to the control, and you see they always have a higher photosynthetic performance with respect to the control, both in leaves that are dark adapted or in leaves that are exposed to light. So we map this mutant by using a strategy that we developed within the frame of the BarPlus project. In this case, we decided to go for exon capture and exon sequencing. We develop an F2 mapping population. We collect 50 plants with the AS1 phenotype and 50 plants with the control phenotype. And in the end, we went to look at the allele frequency and we mapped the mutation in this region on the long arm of chromosome 4H. There were a few candidate genes here, but at the end, we ended up with a premature stop codon in these genes where a tryptophan is replaced indeed by stop codon. We had a perfect Mendelian segregation, meaning that this is indeed a monogenic recessive allele, and in particular, this gene is encoding for a protein that is showing high homology with an Arabidopsis protein that is known as senior recognition particle 43 kilodalton. This protein is a chaperone. It's actually part of an heterodimer, interacts with another protein that is known as a senior recognition particle 54 kilodalton, and they have actually the role of taking the antenna proteins into the stroma of the chloroplast. Usually these antenna proteins are synthesized in the cytoplasm. They are important towards the tick-tock import machinery of the chloroplast. And once they are in the stroma, since they are highly hydrophobic, obviously, they have been taken by this heterodimer and they will be applied loaded on the thylakoid membranes, and they will form the antenna proteins of the photosystems. So basically, here is the mutation that we have isolated. The protein, the AS1 mutant, is missing these two, the C-terminal. In particular, this region contains two chromodomains. Just to prove that we have isolated the correct gene, the corresponding mutant in Arabidopsis had indeed actually the same phenotype, was a pale green phenotype. And they behave exactly the same. For instance, we run some Western blot, you can see the mutant of barley miss specifically this antenna protein, the mutant of Arabidopsis miss the same protein. What happening there essentially is lacking these two chromodomains. The heterodimer in the mutant cannot be formed. The heterodimer in the wild-type sequence is nicely interacting by this BFK into the chloroplast. And you see when we couple the mutant version of SRP43 with the wild-type version of SRP54, we do not get the interaction. Essentially, the heterodimer cannot be formed. These plants have been cultivated. They've been cultivated in Poland and in Spain, and we under standard condition, under standard density condition, and we see that essentially they behave very similarly to the control. So at least what we can say under standard condition, that being pale green does not cause any penalty on yield. It will be obviously very interesting now to verify whether cultivated at high density, they might result in an higher productivity, as has been shown, for instance, for tobacco under greenhouse condition. Now, we were wondering, is any kind of pale green mutant characterized by this reduction in chlorophyll content and a higher photosynthetic yield suitable to these purposes? And then we came to the mutant that we have isolated in the Tilmor population from Silvio Salvi at the University of Bologna. Again, we got a mutant. This was paler, more or less the same content of chlorophyll, although here we show an higher content of chlorophyll A with respect to the chlorophyll B. And again, the same characteristics. It showed a higher photosynthetic rate with respect to the control at all the light intensity that we tested. But then it came as a surprise when this mutant, again, under greenhouse condition, is exactly like the control plants, although it's paler, but for the rest, nothing changed. If you saw this in the springtime, this is in the Morex background, there is no problem. It grows and it produces very similarly to the wild-type plants, supporting the fact that being paler does not create any disadvantage. But if you saw, as we have done last year, in end of October in Milan, in our botanical garden, where we had a very cold winter time, this mutant tends to be totally yellow and then die. So it has a very high sensitivity to low temperature, and we have also tested in the lab by adapting these plants under 4 degrees for a few weeks, and you can see that at the base of the leaves, essentially the chloroplast stopped to develop. You have just a few thylakoid membranes and a large accumulation of lipids into the plastoglobules, whereas at 18 degrees, nothing happened. The chloroplast biogenesis can run without any problem. We went to map the gene. This time we use a different strategy that was developed together with Laura Rossini and David Horner at the University of Milan. This time, instead of exon capture, we decided to go for an RNA sequencing approach. So we analyzed the RNA sequence approach comparing wild-type and mutant plants, but at the same time we also generate a mapping population. We generate 2 bulks with the mutant phenotypes and bulks with the wild-type phenotype, and then we sequence the RNA. We went for variant calling, comparing the results with the differential gene expression, and come to the gene. This time the mutation is located on chromosome 7H, and you can see here the few SNPs that are highly abundant in the pool, in the mutant pool, and they are consequently reduced in the wild-type pool. We had a few candidate genes, And our best candidates is this gene here. The gene that we have found encodes actually for subunits of the magnesium chelatase is the enzymes that takes protoporphyrin 9 and transform into magnesium protoporphyrin 9 and then goes to the chlorophyll A and chlorophyll B biosynthesis. These subunits is actually part of a larger complex. It forms an homohexamer that together with another homohexamer formed by the CHLD subunits and then the monomer CHLH, they interact together and are actually forming the magnesium chelatase. The mutation that we have identified here is actually a nucleotide, is a SNPs also in this case, that replaces a highly conserved arginine residue into a lysine residue. And by modeling the structure of the barley subunits based on crystal structure of the same enzyme in Rhodobacter, we realized that actually the amino acid substitution that we have here is actually a very important substitution because here is indicated in red, is actually responsible for forming those homodimers that together then they form the hexamer. So we believe that this mutation is going to destabilize these homohexamers that actually is important for the ATP hydrolysis during this reaction, and therefore the enzyme activity is reduced. Now, we were wondering, but why our AS1 mutant, so the one affected in the chaperones, does not have this higher sensibility at low temperature, whereas the other one, it has. The answer comes from a study that has been recently published by the group of Bernhard Grimm at the University of Berlin in Germany, where they were able to show that these subunits, the one that we have isolated, that the one that has mutated in AS1, besides controlling the import of the antenna proteins on the thylakoid membranes, they also control the aggregation state of this enzyme, is the glutamyl tRNA synthetase enzyme, that is actually one of the first enzymes in the biosynthesis of the tetraperols and of the chlorophylls. So in other words, if you don't have this protein, on one side you don't upload, or you upload with less efficiency the antenna proteins, on the other side you block the biosynthesis of chlorophylls. The mutant that we have in the MOREX instead is just hampering this enzyme. You reduce the activity of this enzyme, and you will have an overaccumulation of all the precursor of the tetraperol biosynthesis. These precursors are able to absorb light. This precursor will generate a lot of oxygen radical, a lot of oxidative stress, and that's actually what we observe at cold under low temperature. So basically, the pale green phenotype might be a trait to be introgressed into new breeding programs, but not all the pale green mutants are the same. So probably the gene encoding for SRP43 could be a good candidate. I will not touch genes encoding for enzymes of the tetrapyrrole biosynthetic pathway. As I said, this is just part of a larger work that is trying to bring photosynthesis from an artificial environment and from an artificial plant into the real world. And so if there is the wish to have a thematic group on this topic, on chloroplast biology within the barley community, please take us into consideration. I would like to finish to thank, as I said, the AS1 story started within the frame of the Barbara Post project. I would like to thank, obviously, Agnieszka Janak, Laura Rossini, Gustavo Slefa, because of the different contribution to the AS1 project. And obviously, I would like to thank also the CREA with Luigi Cattivelli, Alessandro Tondelli for giving us access to many of their resources. And also, as I said, Silvio Salvi and Serena Rossignoli for the Tilmore population. With that finished, I thank you for your attention and I'm pleased to take some questions.
Yeah, thanks. Very nice presentation. Can you explain why the HAS1 mutant with half the chlorophyll has a better photosynthesis?
Yes, because essentially the leaves absorb a reduced amount of light, and this reduced amount of light reduce the activation of a series of mechanisms that are called photoprotective mechanisms. Among them, there is the non-photochemical quenching. When they are activated, the efficiency of photosynthesis drops down dramatically. If you measure the photosynthesis of a dark adapted leaves, it is always much higher than the photosynthesis of a light adapted leaves. And that's actually what happened in there. Less light, less photoprotection, higher photosynthesis.
Yeah, thank you for the introduction of this nice mood. Have you considered high temperature experiment with these plants? Do you expect opposite phenotype of cold stress phenotype what you have seen for these Hus mutants?
At the high temperature?
Yeah. So what do you think if you grow these plants under high temperature, they do better than the control experiments and gives higher yield because of the pale green?
No, I think here the limiting factor is light. I don't think it has much to do, at least in the case of US1.
Yeah.
In the case of the other mutant, the Tilmore 2490, probably increasing the temperature, but not the maximum temperature, the minimal temperature, as we have shown. If we start growing these in springtime, at least in Italy, it does not have any effect. I don't think it's a matter of maximum temperature, it's a matter of minimal temperature.
Okay, yeah, thank you.
Thank you, Klaus, for the nice presentation. And thank you for the organizers for the invitation. It's a pleasure to be here. It's my first barley genetics meeting, and I'm learning a lot. So I come from Ireland, and when we think about Ireland, some of you may think about the Irish pub, of course. A couple of you also would probably think about Guinness. And probably a handful will think about Irish whiskey. Irish whiskey historically has been for 800 years, and in the 19th century accounted for 60% of the global consumption. So it was very important, but then decreased due to historical motives. So there has been a revolution in Ireland in terms of the Irish whiskey. It's called the Irish Whiskey Renaissance. So in 2010, there were 4 distilleries. Today, there are 32. So it's incredible. And all of this, of course, requires barley. And more than that, because you have so many distilleries, there is a seek for premiumization. What can you have that distinguish your product from the others? So, for example, this paper just came out recently from the Waterford distillery on the impact of terroir and how can that change the whiskey. So, but as Klaus mentioned, now in Ireland, waterlogging and flooding is increasing. So if you think about the Irish weather, this is what happens. These are the seasons of Ireland. And there's a saying, if you don't like the weather, wait 20 minutes. Because it will change. So this is an image of the Waterford distillery in 2019. You can see here there was a rain during, so this is spring barley, of course, and during late March there was a rain, and of course nothing grew afterwards. So for those of you who are not familiar with the term, waterlogging means that we have water on the root level. If you go to partial or complete submergence is when you have stem or even the complete plant is underwater. And of course, hypoxia, so this is the key part, is the oxygen levels in the soil can decrease or even become inexistent. So this is just a quick image just related with the effects of waterlogging on an hypothetic barley plant. So this is what you observe. You have imbalanced nutrient uptake, reduction of root architecture, branching yield, biomass, etc., and the classic erenchyme formation as a strategy. So how am I talking about it? So when I arrived to Ireland, I started thinking about a project that made sense, and I love genetic diversity. So in collaboration with the James Hutton Institute and with the team of Robbie Waugh, we came up with a collection. And what we are trying to think is that modern cultivars, our underlying hypothesis has been mentioned today, this morning, that modern cultivars were bred under optimal conditions. And they are favoring yield-related traits. However, heritage germplasm might have stress features, but it remains largely uncharacterized. So we assembled what we called the European Heritage Barley Collection. So this is just a glimpse of the spikes, how diverse they are. And so the collection comprises 360 accessions, 2-row spring barley. It was collected from, again, as I mentioned, in collaboration with the James Hutton from several different projects across the years. And then we also try to focus more on the Northern European side because, of course, that's where we would find more traits of waterlogging, and as we even went to the Norwegian germplasm bank and multiplied the seeds to go to the field using speed breeding. So we collected this, and the first thing is that we genotyped everything with the 50K SNP array. We can see that the population structure shows 5 groups, and then we did a PCA analysis. So these are undergoing results, so this is quite new. So it was interesting because, as I mentioned, we had seed bank, we went to germplasm banks and all of that, and something that we found is that although it was labeled as 2-row, it was actually 6-row, and fits nicely here in this group. The genetic and, of course, the agronomic data also clarify that. But there's no distinguish, and then we try to see, do we have a difference in terms of year of release? A previous paper that we worked with Stephanie Saad, we observed that there was a sort of a line between the cultivars before the 90s and after the 90s. And apparently this is what we see here in this PCA. It seems like we have the land races in green, so all of these, they are in this area, and then you have the cultivars mixed between after the 90s and before the 90s. And then we try to see, do we have any geographical difference, but apparently it seems like the whole groups are converging together. But again, we are still analyzing these results. So, okay, 365 lines, we have land races, we have formerly bred cultivars, but there's still a lot. Especially to go to the field, and so we established what we call the core collection. We did a power analysis, and we went from 365 lines to 230. So the first thing that we did is that, of course, we went to the field. So this is the UCD Lions Farm, located in Kildare, which is a little bit up northern Dublin. And so we looked at several traits: shoot biomass, yield per square meter, harvest index, height, 1,000 kernel weight, heading date, etc. And what you see in gray is the exhibit collection of the 365 lines. And what you see in yellow is those 230 core collections. So we can really capture not only the genetic diversity with the SNPs, but also the agronomic diversity. So this is all very nice, but how does the exhibit respond to waterlogging? So we went to the field. If I could go back in time and say to myself 4 years ago, I would say, don't do it. because it was really the logistics is crazy. So we usually sow in Kildare because of the temperature of the soil. We sow a little bit later. So we sow usually by end mid-April. By the middle of May, we have the field preparation and the irrigation set up. So we use pipes, and and then by the end of May, we normally do the stress imposition, which means that the plants are early tillering stage. We do data collection throughout the season with both drones and also agronomic data. So, and then by end of August, we harvest. So we started, the first year was in 2020, it was extremely challenged, as you might understand, because of COVID. So we started by testing how much water do we really need to waterlog the field, how many days do we need. So we did a simple test pilot. This is just a drone image, and then we measured the amount of water in the soil and estimated how much water we needed. So we thought, okay, 5 days seems the soft spot, so it looks good. And we designed a field for 2021 that with the 230 lines. This was just 3 cultivars just to test. It was optimization step. And we went to the farm manager and we said, okay, we want 230 lines. We have to do the account for replication. We need 2 hectares and we need lots of water. And the answer was no, the cows needs to drink the water. And so we had to redesign the field trials to make them smaller. So that's what we did. We got much smaller field plots, so it's 1.5 meters by 50 centimeters, so quite narrow. But as you can see, we have 3 rows, 15 centimeters apart. But even though the logistics, we had to come in with large tanks, water pumps to pump up the water from the mainstream, generators, and to get all the water. So what we did was that we only did 4 days of irrigation at early tillering stage, 12 hours per day, and that was already more than 250 cubic meters of water. And this is what you can see here. This is just a soil water content. And if we zoom in to these first days, we can reach the saturation around by the end of the first day. And it was also a very fun fact is that it was pouring rain and we couldn't install the irrigation pipes. We had to delay the water logging because the team could not go to the field because it was raining so much. So it was very mushy, and this year again, it was raining and raining. So we not only use the probes in the soil, but we also take a core sample of the soil, and the core sample is like chocolate mousse. It's totally mushy. So this is 2021 data. We are now running our second year of field trials. So as you can see, there's interestingly, the waterlogged plots flower later. The plant height is reduced and the shoot biomass is reduced. And this year I was with the visitors with some visitors in the field, and they were very interested in an Irish heritage line. And we went to the control plot and looked nice and neat, and we went to the waterlogged plot and it didn't look very nice at all, and it was still not flowering where on the control plot it was way more ahead. So, but also interestingly, we also try to see what about the grain quality. So we looked at the protein content. Again, under waterlogged conditions, you have a big reduction of the protein content and also the beta-glucans. So we are in the middle of our project, so it's still a long way to go. And we are also looking into drone imaging, so we are flying drones in the field. And this is just an image of how does the field look like, and before the waterlogging and after the waterlogging. And we are processing the data, and the aim is to use machine learning and artificial intelligence to analyze the features and try to recognize the best predictors of waterlogging stress, because this is a longitudinal study, so we have several time points, at which time point is the most interesting feature. So image-based phenotyping has some challenges, as Sergey mentioned. So, but also doing waterlogging in field conditions has lots of challenges: the logistics, the cost, the water availability, leveling of the fields. We can only do the imaging with UAVs because the field is so mushy that you cannot have a ground-mounted sensor. And because we have the irrigation equipment, as soon as we finish the waterlogging, we take all the irrigation, because then we have unwanted objects that will affect the machine learning. So, of course, UAV's challenge, I could talk about that for a while. And, of course, all the ground truth that we require. So we are collecting also leaf area index, all the RFS data, and weather data as well. So what we are doing is that we are analyzing the drone data, and we are using machine learning to interpret it. We are also imaging the same 230 lines of the core collection in controlled conditions in collaboration with UPGV, Université Picardie Jules Verne in Amiens. And we are doing this field trial this year. And the take-home message is that we have this exhibit collection, if you want to talk to me about it. It's very genetically diverse, and the idea is that this is a resource for the community. It can be used for multiple purposes. I didn't mention here because I could not cover everything. We have also looked at the root, and the root architecture is radically different from the nowadays lines. It's a much bigger root architecture. And waterlogging impacts agronomic and quality traits of barley. And we are flooded with data, and that's why we are using machine learning. And so I would like to thank, this is a big team project and collaborators, not only from the UCD, the people behind. Most of the things that I mentioned was produced by my PhD student, Vilo. Vilo, she will give a flash talk today, and she also has a poster. But also the James Hutton Institute with Robbie, Joanne, Callie, and Luke, and also the UPGV, and of course the funding from Science Foundation Ireland as the President Award. And thank you, and I welcome any question.
Thanks. A quick question. Waterlogging caused two major constraints. Hypoxia is one of them, and elemental toxicity, increased content of manganese in iron due to redox potential change in the soil. So which one you believe is more important here? What was the soil type?
That's a very good question and hard question. I would say that one of the things that we want to do, we are still not doing it because of the amount of the field trials, but we want to now reduce probably to a couple of discriminant lines from the exhibit collection. And we want to look more carefully at the soil composition and also the water composition, the water source composition, because that's also very important. So we can talk in 1 year or two, Sergey.
Yeah, sure. Thanks.
Sonia, thank you for your presentation. I have two questions. The first one is why you choose this particular phase to apply the waterlogging, because normally it's when occurring the waterlogging there in your area. This is the first question, because the most critical period for waterlogging is close to flowering in general, more than during the tillering. This is the first point. And the second is the DPV has very important effects in waterlogging, depending if you have more DPV demand from the atmosphere or not. And what do you think if you apply the same treatment in a hotter condition, another year, for example?
Well, we are doing it in controlled conditions, so in the glass house where we have everything very controlled. Could you repeat again the first question? Sorry.
The first question is why you choose this particular phase of the crop to apply that.
Yes. So at early seedling, so it's all related with the farming practices in Ireland, right? So when the rain occurs more is usually can be in the springtime. And in the springtime, at this stage, that's what happened on that distillery. The plants are in a early seedling stage or something like that. So it's when the adventitious roots is starting to form. So the plants are quite sensitive at that stage as well. And at the early tillering stage is when you have the tillings forming and also the little tiny spikes. So that's when we also expect that the yield will be more affected. So it goes because that's what happens in nature. But I mean, I'm saying this, but of course we are seeing all changing. So for example, in 1 year we can have flooding at springtime and then drought in the flowering stage. But in 2020, when we were collecting the flowering time of the big exhibit collection, there were storms, 2 storms in the middle of July. With massive winds and a very high rainfall. So the historical data is changing a lot. So, yeah.
Thank you. Sonia, can we learn anything from rice? Is there the submersion tolerance mechanism in rice well understood? And do we know whether similar genes pathways are present or absent in barley?
There's lots of, I didn't mention this, but there's loads of studies, of course, in waterlogging for many years. And of course, in Arabidopsis and in rice, Julia Bailey-Serres has been done lots of work on it. And the molecular mechanisms are, are very well understood. You have the ethylene responsive factors, the ND-GRON pathway. So all of the fundamental behind it has been very well studied. So in rice, you have the ethylene, of course, is a main regulator. And you also have the main structure, which is the erenchyma. So that's why. So one of the things is how barley produces the erenchyma as a strategy to cope with the stress. But rice has, it's there. The erenchyma is always there. Yeah.